Hi everybody, it is your AP Biology teacher, Mr. Poser. Today we are continuing our Unit 7 recap with our Part 2, where I'll be covering topics 7.9 through 7.13. So Unit 7 is the biggest unit and is probably the most important, intensely covered on the AP exam, so that's why we're splitting this into two. So let's just get into it right away. So the first part of the video was all about how natural selection works, what evolution is, what it looks like when it is happening, what it looks like when it isn't happening, and how we can use math to tell whether or not it is, and all the evidence that we have that species in fact do change over time and species do come from other species. So to put all that together, we can put it on a diagram, and that's studying a phylogenetic tree. So this is the study of phylogeny, the evolutionary history of a species or a group of species. In a diagram like this one, this is the tree of life, kind of like what Darwin drew in his notebook. It's a diagram that shows the evolutionary history based on kind of the forms of evidence that we've talked about, like a fossil record, comparative anatomy, especially DNA and molecular similarities. So something that you're going to see on the AP exam are some phylogenetic trees, and we got to learn how to read them and figure out what they mean. They are a lot like family trees. They help us to determine where species came from in regards to what species they descended from and how closely related they are to each other. And like I was saying, evidence like morphology, body structures, and molecular data are the main two sources of information that are used to build these phylogenetic trees. What do they have in common? And the more they have in common in terms of their body structures and in terms of their molecular similarities, the closer they're going to be together on the tree, and therefore they're going to have a more recent common ancestor, right? They would have evolved from the same species sooner. So what we call these deviations on a phylogenetic tree, where we can see two species or two groups of species branch off from each other, that's called a branch point. Makes a whole lot of sense. And those are representing divergence of two groups of species from one common ancestor. So here's a branch point between Chiroptera and, well, pretty much all these other groups down here. Here's another branch point between Perissodactyla, horses and rhinos, and these two groups, Carnivora and Phylodota, or Pholidota. I don't know all of these by heart, but those are branch points, and those are representing basically common ancestors of groups of species. Two groups of species or two species that are closely related to each other, and they have an immediate common ancestor on a diagram. We call them sister taxa. And then the basal taxon that taxon is singular, taxa is plural. The basal taxon is a group that diverges early and it's separate from all the others on the tree. So this is a tree of mammals, okay, and it makes a whole lot of sense that monotremes are the basal taxon of mammals because they lay eggs. Yeah, so this is platypuses and echidnas and stuff, and they diverged from the rest of the group the earliest, and that's why they were common ancestors so far back. Okay, so the common ancestor of all mammals is back here, and that's that branch point. Okay, to phylogenetic trees show relative age based on divergence. So generally speaking, the further back you are on the phylogenetic tree, or further like up the tree or down the tree, it usually is, wherever you see more of the branch points, the deeper branch points, those are going back in time. So this one, we can assume that time kind of goes this way, and these are the modern species, and then the branch points are the more ancient species. So we can say this one that I'm kind of highlighting right here is more ancient than, say, this one or this one. They imply evolution from a common ancestor, and it's not what's called adjacent evolution, right? So one of my big pet peeves about people's misunderstanding of evolution is that, oh, people came from monkeys. Like, no, first of all, we have a common ancestor, a recent common ancestor with apes, right? And those aren't monkeys either. So adjacent evolution would mean like, oh, these, let's see, these armadillos evolved from elephants and that kind of thing, and that's not accurate. But it shows that they have a recent common ancestor, and that's something that you got to know as a biology student, as a high school student. They show patterns of descent, not necessarily similarity in features, right? So even though two species might look really similar, they might not be closely related, and that might be because of what's called convergent evolution, where two kind of distantly related species evolve similar traits because of living in a similar environment. I'm going to put that in here for a sec. Hang on. That's what we call convergent evolution. All right, so they converge, right? They come closer together. So therefore, molecular evidence is the more reliable form of evidence for phylogeny. We can basically determine their molecular similarities or molecular homology. What percent of their genome is the same, or how many genes do they share, how many amino acids in their cytochrome C do they share or do they have in common, and that's more reliable for building phylogenetic trees. So another kind of branch of phylogeny is what we call cladistics. And this is something that you might also see on the AP exam, something called a cladogram. And it's a classification based on grouping organisms into clades, or groups that include ancestral species and their descendants. And cladistics, a cladogram, is based on homologous characteristics, or what we call shared characters. So this is kind of more like what a cladogram looks like. And a cladogram on the bottom of the tree still represents the common ancestors, the branch points, and then these are the modern species. Speak of the devil, look at that. Apes and humans share a very recent common ancestor. So if you want to look more into how to build a cladogram and how to use a cladogram, I suggest going to, I believe that would be 7.9 video that I have on my channel. But for now, since this is a recap, we're just kind of going for it. So we're going to go to 7.10, and this 7.10 is all about speciation, and that's a form of what we call divergent evolution, where two species become more different from each other over time. They split apart evolutionarily. They might share a recent common ancestor, but then, speciation is where two species have a common ancestor and then they become two separate species. That's the formation of new species. And how do you get new species from an old species? Well, two groups become what are called reproductively isolated, meaning they cannot produce viable, which means the offspring can't grow, and fertile offspring, which means in order for two individuals to be a member of the same species, they need to be able to produce babies, and those babies need to be able to have babies as well. And two organisms are not members of the same species if they cannot do that. That's what we call the biological species concept. So here's a very, there's a thin-lipped cichlid and a thick-lipped cichlid. Thick-lipped cichlid. Say that 10 times fast. And they share a very, very recent common ancestor, right? One of these species more than likely diverged from the other one. And if they can't reproduce viable fertile offspring, therefore they are not the same species. So here's a very general overview of how new species form. You have one population, and then they become separated somehow. There's lots of different ways that they could become separated. We'll talk about that in a sec. They evolve separately over generations, and they adapt to their environments through natural selection. And then they are what are called reproductively isolated, that even if you brought them back together, even if they are no longer separated, they are still two separate species because they cannot produce viable fertile offspring. That's the general way of how speciation happens. Okay, and something I already mentioned, the biological species concept. A species is a group of organisms who can interbreed and produce viable fertile offspring in nature. If they can't do that, they are not the same species. So what I have here, this little cutie, is a liger. It's pretty much my favorite animal. I hope you understand that reference. It is a cross between, I want to say, a female lion and a male tiger, I want to say. But it is a hybrid animal, and hybrid animals are interesting because, they are the product of two animals from different species, but they are infertile. They are sterile, which means that they cannot produce, baby ligers. It could be because of a chromosomal difference. It could be gametic incompatibility, a whole bunch of reasons. But therefore, a liger is not a species. Lions and tigers are very closely related, but they are not the same species. So something that might prevent a species, two individuals from producing viable fertile offspring and thereby separating them as species is what we call a pre-zygotic barrier. And hence the name, it prevents a zygote from forming, pre-zygote. And remember, a zygote is a fusion of cells between a sperm and an egg. So here's some examples of pre-zygotic barriers. There's geographic isolation, where there's a physical separation of populations by some kind of geographic barrier, a river, a mountain range, something like that, maybe a city, I don't know. That's geographic isolation. Temporal isolation, separated by separation of a population into two or more based on mating or breeding periods. And then 3, their behavioral isolation is separation of a population by mating behavior. They no longer recognize the mating habits of the other, based on this picture that I have over here. I don't even know where this came from, but it does a pretty good job. But those are what we call pre-zygotic barriers. Those prevent a zygote from forming. And hang on a second. Oh, yeah, there's 2 more. I was like, wait, I'm on the wrong page. But no. Pre-zygotic barriers, 2 more, are mechanical isolation. 2 groups are too anatomically different to mate. Like, you can piece that together, right? Their parts will not allow them to produce new species, right? Makes sense. And gametic isolation, the gametes of the groups are incompatible, and therefore they cannot fuse and form a zygote. Okay, so your 5 pre-zygotic barriers: geographic, temporal, behavioral, mechanical, and gametic isolation. And the main point there is that those are preventing a zygote from forming. But there are a few post-zygotic barriers as well, okay? Because remember, the biological species concept says you have to have a viable, fertile offspring. So here's the deal: the offspring could, the zygote could form, but the offspring might not grow into another animal, or it might not grow into an adult, and it might not be fertile. So here's some post-zygotic barriers. They prevent zygote development into viable, fertile adults. One example of this would be hybrid breakdown or inviability, meaning that the zygote cannot successfully grow into an adult, and then reduced hybrid fertility if the hybrid cannot reproduce. So here's another good example. I believe this is a growler bear, and I think that's a real thing, a growler bear. I want to say, again, it's a female polar bear and a male grizzly bear. I might be wrong on that, but you get the idea, right? It's a growler bear is not a species, and that thing is going to be basically infertile. All right, speciation happens. We categorize it in 2 main ways. So remember, we had a population that becomes split somehow. They evolve separately, and then they become reproductively isolated. This is the 2 main groups of speciation that we have. If speciation occurs through geographic isolation, basically if they're separated from each other, that pre-zygotic barrier, that's what we call allopatric. And then sympatric speciation is speciation that occurs in the same area. And there's some other pre-zygotic or post-zygotic barrier that is preventing the species from, two groups of organisms from being the same species and producing viable, fertile offspring. All right. So yeah, this is allopatric, right? Geographic barrier, reproductive isolation. Yep. And here is sympatric, right? It's the subgroup of the population, grows within the same geographic area as the first population. There you go, that is sympatric. As I mentioned before, speciation is a form of divergent evolution, and that makes species more different over time. And what I mentioned at the beginning of this video, talking about why molecular similarities are the best evidence for phylogenetic trees, to build phylogenetic trees, is because of convergent evolution. So these 2 are opposites. Species become similar over time due to their being in similar environments. Okay, so this kind of fish shape adaptations, that's an example of convergent evolution, because a mammal is not very closely related to a shark, but it kind of looks like a shark. Well, a dolphin kind of looks like a shark, right? And an ichthyosaur kind of looks like a shark too, but it's not. It's a reptile. And so that's what's convergent. Okay, divergent evolution, the kind of evolution where they become different over time. The speciation being the main example of that, can occur via what we call gradualism, where small gradual changes over time cause species to become different from each other, or through punctuated equilibrium, where there's a long period of very little evolutionary change, and then there's all short periods of rapid, really quick change, depending on, once again, the environment and natural selection. So yeah, gradualism and punctuated equilibrium, those are kind of 2 opposites when it comes to divergent evolution. And there's a lot of terms to remember here for patterns of evolution. So divergent evolution is species becoming different over time. Speciation is the formation of new species. The opposite of that is extinction, the elimination of a species. And this opens up a whole nother can of worms. A species is extinct when it no longer exists or cannot reproduce on its own. And here's a fun fact: 99% of all the species that have ever lived on planet Earth are now extinct, which means all the forms of life that are around today make up only 1% of everything, all the varieties that have lived on planet Earth, which is amazing. So extinction, what it does, it reduces the planet's biodiversity, the diversity of living things. So that's a pretty huge deal, and that's shaped life on Earth in a very, very, very big way. So extinction rates are high during great ecological stress when the environment changes drastically. And when we have a very high rate of extinction, it's what we call a mass extinction. And mass extinctions result in large amounts of species going extinct at the same time, very rapid change, and but extinction is a normal part of the history of life on Earth. Species go extinct, make ways for better adapted species to a particular environment. The environment changes, and then the species that were adapted initially are no longer adapted, and a new species might take their place. That's a normal part of the evolutionary process. That's just life. Literally, it's life. That's what we call the background extinction. But this picture down here definitely is a mass extinction, right? So the Chicxulub crater is the evidence of a giant asteroid, wiping out the dinosaurs. Although there's new evidence that might say it wasn't the asteroid that killed all the dinosaurs. So. Okay, and also you might not say that dinosaurs went extinct in the first place because they evolved into birds, right? And there's the theropods, right? But here's a quick graphic of the 5 major mass extinctions that Earth has had in the last 500 million years. I'm not going to read through all this, but it's just interesting to know. It's good to know, and we're going through a 6th mass extinction right now. So PSA, species are going extinct really, really fast today. As I was saying, though, right, extinction is a normal thing. Extinctions enable new species to evolve. They fill new niches or the roles of eliminated species. And it changes. Environments change, species change, biodiversity changes. And extinction is essential for the continuation of evolution. Like mammals wouldn't have ever diversified in the way we have if it weren't for the dinosaurs kind of just going extinct. So as I said, it shaped the history of life, and extinctions shape evolution. So here's a quick definition of biodiversity, because this is a huge concept, especially for the rest of the Unit A and for Unit 8. It's the diversity and the variety of living things, and that's impacted by both speciation and extinction, right? So biodiversity will go up if speciation is occurring, or if speciation is greater than the extinction rate, but it'll go down if speciation is smaller than the extinction rate, or the more species are going extinct faster than new species being formed. Okay, biodiversity is a super important concept because biodiversity accounts for the genetic variation of a single population as well, and that's a super duper important thing. We talked about this in 7.2, how natural selection cannot happen without genetic variation, and that's the main key here, is that genetic variation is ultra important. So going back all the way to the bugs and the birds example that we had, like if all those species, or all those members of that species, are the exact same, nobody's going to have an advantage over anybody else, and therefore the population cannot adapt, and it cannot grow, it cannot evolve. So genetic variation is ultra important. So as I just said here, populations and species cannot adapt or respond to environmental changes if there are no traits that enhance reproductive success. If everybody's the same, nobody's going to be better suited for the environment, and therefore natural selection is not going to happen. So therefore, species with low genetic variation are at a greater risk for extinction. So 2 things about that. That's why we kind of refer to species endangerment as kind of like a vicious cycle, right? Because once there's very few of them, there's very little genetic variation, and once there's little genetic variation, they're at even higher risk of going extinct in the first place. So this is interesting. This is what I think it's a whiptail lizard, it's called. And this entire species, this entire population, I believe it lives in like New Mexico or the deserts, in the western United States deserts. They're all females. They're all clones, and they just lay eggs, and it's more of the clones. So there's very little genetic diversity there, and I'm not entirely sure how that evolved. It must have been an advantage at some point in this species' evolutionary history, but I mean, they could be at risk for extinction, right? Because they have 0 genetic diversity. So yeah, once again, a higher genetic diversity means a more resilient population. It's more able to adapt to changes in the environment. More variation increases the chance of a advantageous gene or a trait. It is crucial for withstanding environmental changes. And here's another example of a at-risk at-risk organism here are bananas, right? Bananas are all one variety, and they're all basically, all the banana trees are basically clones of each other. They're all one kind. So like if there's a banana blight, like that's the world's fourth largest crop, you might be screwed. So as I was saying, the main idea for this is that genetic variation is key when it comes to biodiversity and when it comes to adapting to environmental changes through natural selection. The same goes for ecosystem biodiversity. That's another kind of biodiversity that's super important. And the common theme here is that it's something that we've talked about in a previous topic in this class as well, is that the more variety, the better. And here's why. Again, an ecosystem like a rainforest has a whole lot of biodiversity and is therefore there's a higher chance of a species in that ecosystem being more resilient to environmental changes and having an advantage. It's the same principle that we see over and over again. So think about the biodiversity in a rainforest, which is one of the biodiversity hotspots on the planet, versus that of, say, a cornfield, where it's all one type of plant, and often these times these plants are all clones of each other. So there's virtually like zero biodiversity. Which one is going to be better adapted to environmental changes? It's going to be the rainforest, the one with more biodiversity. The same principle applies. So 7.13 is the very last topic of Unit 7, and it is about the origin of life on Earth, which is an extremely interesting topic, in my opinion. We don't know the answer to it yet. We don't exactly know with certainty. Well, we don't really know anything with certainty, do we? But we don't know for sure how life began on planet Earth, and it's a really intriguing concept. So we're going to cover some of the basics here in this recap, and at least the hypotheses that are covered on the AP Biology exam. But how I'm going to talk about it here is I made another very simple flowchart of what had to happen in order for life on Earth to begin. And that's inorganic molecules, the ones that appear all over the place in space, would have had to form organic molecules like, say, amino acids and carbohydrates or simple sugars, fatty acids, nucleotides. Somehow inorganic molecules had to have become organic molecules, the kind that you would find in a living thing. And then these organic molecules, think of monomers, these monomers would have had to become complex polymers to form proteins and nucleic acids and complex carbohydrates and lipid membranes, that kind of thing. So this two jumps so far, one from inorganic to organic and then organic to complex polymers. Hey, and somewhere down here those complex polymers were able to assemble themselves in a particular way that lends itself to a living thing. It's able to reproduce and pass down genetic material. It's able to maintain a stable internal environment, metabolize, exchange materials with its environment, respond to stimuli. Somewhere in there that happened, and we're not 100% sure how that happened yet, and we're not really 100% sure about how life began at all. So here we go. Let's talk about what we do know, though. All right, so one of the things that we do know is that Earth is about 4.6 billion years old, and that is unfathomably a large number. It's a really, really tough to conceptualize, tough to wrap our minds around how long of a time that really is. And life on Earth has been around for, our best estimate is 3.5 billion years. The oldest fossils that we have found are about that old according to our radiometric dating data. And that's something I spoke about in the last video, is when you have a sample or a fossil like a rock or something like that. There's some radioactive isotopes in there, so basically atoms with too many neutrons. And when those neutrons kind of break off of the nucleus of those atoms that have too many, they go from being an unstable radioactive isotope to a stable one, and the time it takes that to happen is predictable. It's called a half-life. So that's how we're able to figure out that, hey, this living thing, this once living thing, this fossil, first appeared 3.5 billion years ago, and that's when we think life began on planet Earth, which is a very, 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 very long time, which is how we can get these gigantic macroevolution changes. Early Earth's condition was unsuitable for living things, so in the Hadean Eon, as we call it, Hadean being after, I believe, the Greek god of the underworld, so it's like basically hell, right? Ooh, bad word. Here it is right there. That's an artist's depiction of what Earth may have looked like, and no, that is not friendly for living things. But you know, this is this is a baby picture of Earth. After a couple billion years, you know, it got a lot more suitable. And it only took about a billion years for life to form on planet Earth. So go us, right? Okay. But here's my flow chart again. How the first step that we got to talk about here is how we went from inorganic molecules to organic molecules. And one of the most famous hypotheses, explanations for how that came to be is called the Oper-and-Haldane hypothesis, or at least this is the one that's going to be covered in AP Biology course and exam description. So here we go. Here's what it is. Operin and Haldane are two scientists who hypothesized that early atmospheric conditions, so compounds in the atmosphere, enabled the formation of organic, aka carbon-containing, life-enabling compounds from inorganic ones. So take a look at these compounds over here. We got water, which is you know, a super important part of living things, but technically not organic. So water, carbon dioxide, methane, and ammonia. These are containing atoms that you can find in organic compounds, but are not really organic compounds. They're really waste products. We breathe out carbon dioxide. We, you know, eliminate methane and ammonia. But these combined and under the right conditions could yield sugars, amino acids, lipids, and nucleotides using a little bit of energy from lightning or from radiation. That's potentially true. A secondary kind of sidebar hypothesis is that meteorites could have also carried organic matter to the planet, because meteorites have been found to contain amino acids, which is something crazy to think about, right? But here's the thing about Operin and Haldane. Their hypothesis was corroborated. It was pretty well supported by what we call the Urey-Miller experiment, and another set of two scientists, Urey and Miller, demonstrated that under the right conditions, inorganic compounds can form organic ones, which supports the Operin-Haldane hypothesis. Right? And here's just a kind of a simple picture of the conditions that these two scientists combined: some methane, ammonia, hydrogen gas, water vapor, and used a special kind of condition here in order to produce amino acids, and in order to produce different compounds, but amino acids in particular, which is pretty crazy, right? That was able to be a thing. So how did we go? Let's talk about step 2 then. How did we go from organic molecules to complex polymers? There's a couple ideas about how that could have happened as well. How did monomers cluster into polymers? Under some conditions, amino acids can spontaneously form proteins, and this has been shown by a couple different experiments, especially on a particular type of substrate. And one that in particular that's been mentioned is a type of clay called montmorillonite. And on montmorillonite, this substrate, amino acids have been found to spontaneously form, or excuse me, RNA nucleotides. RNA can form from nucleotides on this montmorillonite substrate, which is crazy. That's a genetic material, right? That can be passed down from parent to offspring. And that's what life does. So that's pretty crazy to think about. So basically what we're saying here is that under some conditions, amino acids can spontaneously form proteins by themselves, and nucleotides can spontaneously form RNA by themselves, which is, you know, that sounds like a really, really big important step in forming a living thing. Now, when it comes to those molecules forming, you know, working together, forming a metabolism, forming genetic material that can reproduce. Here's some of our ideas about that. It says organic molecules tend to cluster, and lipids tend to form vesicles. So think about a bubble. We do a lab in my AP Biology class where we pretend a bubble, like a soap bubble, is a plasma membrane, and we observe the properties of the plasma membrane using the bubble. And the thing about a bubble is that it is remarkably close. The compounds in a bubble, the lipids that you can, you know, find in soap that help you, you know, break down grease off your dishes, those lipids tend to be very, very chemically similar to a phospholipid, and therefore they kind of form a layer and form a bubble. So that's what we kind of think was the first living thing. It was basically a bubble with some self-replicating RNA and some self-contained reactions, which is, your first membrane and your first metabolism and your first genetic material, and it's what we call the protocell. That's what we believe maybe was the first living thing. Just crazy to think about. So then, okay, we talked about organic molecules forming complex polymers. Now, this last step here, going from complex polymers to living cells, there are two hypotheses that are mentioned in the AP Biology exam about how this could have been. They're called the RNA world hypothesis and the metabolism first hypothesis. So it's like, what came first, the chicken or the egg? RNA or metabolism? That's basically what we're asking here. So the RNA world hypothesis states the formation of RNA as the genetic material initiated life. Okay, so basically, again, this kind of depends on our definition of what a living thing is. If it's just, is it just DNA that's able to copy itself, or RNA that's able to copy itself, or is it more complicated than that? What qualifies it as a living thing? But RNA, self-replicating RNA, was believed to be the first genetic material. And the RNA world hypothesis states that that was first, and that initiated life. So special enzymes called ribozymes made copies of RNA in the very first cells, and have led to longer RNA molecules. And we've seen, and this has been studied, that our ribozymes are able to copy RNA in a particularly interesting way, which makes it possible for copying RNA and passing it down to offspring. So that's pretty cool to think about. And then last thing here in the whole unit is talking about the metabolism first hypothesis. So here's my map of metabolism again. And it's kind of stating that complex reactions occurred in a stable environment, perhaps a bubble first, before self-replicating RNA, and provided the chemical basis for RNA to form and for that replication to happen. So it is what the title says: metabolism came first, or RNA came first. The second bullet point I have says self-contained chemical reactions may have predated the formation of proteins and nucleic acids from amino acids and nucleotides. So yeah, that's pretty much what we got for Unit 7. Please let me know if you have any questions, and we will see you for Unit 8. Bye.